Recording is started.
Okay. So can I start the proceeding?
Yes.
All right. Dear Dr. Deshmukh, Dr. Patankar, ladies and gentlemen, today we have Dr. S.R. Dave with us, who will deliver a talk on a very important and interesting area of microbiology, that is bioextraction of metals from their ores. Now over to Dr. Kulveer Dhinsa, who is the coordinator from DAV College, Chandigarh.
Very warm good morning to all of you. All of those who have joined here, we are really pleased to see, and some more are joining, sir. I hereby extend my warm welcome to President of the Microbiologist Society India, Dr. A.M. Deshmukh, who has joined us kindly. Respected Dr. Swaranjit Singh, all the life members and the others who are joining us. We are in fact privileged to have with us Dr. S.R. Dave, who will be the key speaker for this webinar on bioextraction of metals, a green sustainable technology. It is said in Gurmukhi, Pavan Guru Pani Pita Mata Tart Mahat. So all the scientists, most of the research is for the benefit of the human beings, and saving the environment is the first thing which is very important today. And this seminar, which will be conducted, webinar, which will be conducted today, in fact, will be dealing with the same thing. And this is organized by the Microbiology Society India Chandigarh unit in association with SAS Polyclinic Mohali. In fact, I am honored to be given this opportunity to kindly introduce the speaker, Professor Dr. S.R. Dave. We welcome you, sir, and a short your CV. There is the adjunct professor at JVA Research Foundation, Loyola Center for R&D Ahmedabad, as well as the Gujarat Vidyapeeth. He has been honored as the UGC Emeritus Professor. Dr. Sailesh R. Dave was former Director of the School of Sciences, Senior Professor and the former Head, PG Department of Microbiology and Biotechnology, as well as the Coordinator for Clinical Research Programs of Gujarat University, Ahmedabad. 38 of his students have been awarded PhDs and 50 students have completed their MPhil degrees under his guidance. There was one student, PhD student, who worked with him and has received the prestigious Dr. Vikram Sarabhai Award of Biotechnology for the year 2003 by Science and Technology Department, Gujarat.
I request everybody to mute their mics. Please mute your mics.
After a small interruption, because we had to tell everybody that they should mute the mics, otherwise the voice is not clear, it echoes.
Yes, please mute your mic, all of you. Yes, okay.
After I finish, I will mute them, sir. Sir has worked as a resource person, a teacher and expert for the more than 45 workshops and refresher courses also. And sir has published more than 130 papers in journals of national and international repute. And he's reviewed several research papers for more than 35 national and international journals in the field of microbiology, biotechnology, environmental biology, biodiversity, biogeogro hydrotechnology, etc. There are more than 350 presentations in national and international seminars on his name, and he's contributed numerous articles and chapters in books. Sir has handled about more than 19 research projects funded by various funding agencies. Dr. Dave is a fellow of the various academies such as FAEB, FGSTA, FISBT, FBRS, and many others. He is the member expert in several committees constituted at the national and international level, and a member of the Executive Court and Academic Council of Gujarat University. Sir was recognized as a PhD guide for 4 universities and has been awarded Hari Om Ashram Award also. Under his guidance, bio-leaching process and the acid mine remediation technology is developed for Gujarat Mineral Development Corporation. Environmental, microbiological, and for food safety problems of several industries have been solved, and the solutions have been given by Sir's team. He has filed 4 patents. 2 of them are already awarded and 2 are waiting for the final decision. We fall short of the words introducing Sir because he has done so much very interesting and important research for the benefit of the community. Sir, we welcome you, and kindly you can start your talk.
Thank you very much. Just 1 minute, I will share my.
पानी है, साफ सफाई होती नहीं है.
1 minute only,
बताए.
sir. My
खतरनाक कौन?
so please mute your mic. I'm requesting all. Please, Dr. Rajesh, please mute your mic. Dr. Rajesh. Dr. Rajesh, please mute your mic. I request everyone to mute your mic. Okay, thank you, thank you.
Okay, shall I start?
Thank you.
Okay, shall I start?
Yeah, sir, you can.
Okay, now I was forgot to ask now what is the time period I can talk. Hello?
Hello, you can talk 40 minutes, 45 minutes,
Okay.
as you like.
Okay. So
Thank
accordingly, I can frame
You can.
my talk. So
It is up
good
to
morning.
you. It is up to you.
Good morning to all of you. Now, dear Professor Deshmukh, Dr. Saranjit Singh, Dr. Patnikars, and many of my colleagues, as well as participants, those who have joined for this webinars. I am very much thankful to the society for giving me this opportunity to talk about the subject and the area which is very close to me and where I have worked from 1977 till date. I am working on this particular topic. So today's topic, what I'm going to talk about, the bioextraction of metal. And as I have mentioned that I have started my work in 1977 when I joined my PhD at KREC at that time, and now it is a National Institute at Mangalore under the metallurgy department. So a biologist working in the metallurgy department, because the topic is mainly having the interdisciplinary subject of extractive metallurgy, biochemical engineering, chemistry, microbiology, and environment science. So today I am focusing mainly on the bioextraction of metals, now as a green technology as well as a sustainable technology. I'm not going to discuss much about the research data, but I will try to go for the basic aspects looking to all the participants. So the content of the talk, what I am going to discuss in this 45 minutes is the introductions regarding this subject, the major organisms which are involved for the bioextraction of metals, major mechanisms which is playing important role for the extraction of metal ions, and some of the selected examples of a bioleaching, why we say this is a green technology, and then it will come to why this technology is sustainable, and then we'll discuss the carry home message from this talk. So as the bioleaching of metals is a very natural process. We know since centuries that slowly minerals are formed. minerals are degrades, sedimentation will take place, dissolution will take place, and we are all familiar with a geochemical cycling of various elements. Among these elements, metals are one group of the elements which is cycling due to the unknown creatures before few hundred years before. And when people have started searching for these small entities which is responsible for extraction of metal in a natural resources such as Rio Tinto mine. Now, this will give the idea that without knowing, without manipulating anything, these microorganisms, they are responsible for extraction of metals, and specifically it was observed for the dissolution of copper as well as solubilization of pyrite into the ferrous sulfate. Hello?
એક બાજુ કેમ મીઠું જાય કરે રૂપિયા આગળ શું કરવાનું
Hello? Okay. No, no. Can you see the slides? So we are going to talk about this natural process, how you can control the natural process, how you can optimize to get the maximum benefit out of this. So this process, initially it was started at mining site for the extraction of metal ion from the ore. So we should know, because many of us, those who have participated in this webinars are biologists, and the technical word ore, that is for a geological scientist and mineralogist. So ore is a typical rock that contain minerals that can be extracted through mining and the refined use. And minerals is a naturally occurring solid material due to the geological processes, which takes thousands of years. For the formation of minerals, thousands of years will require, and from these minerals, after the geological survey and estimations of a metal ion present in the particular place, the specific ores are mined and they would be analyzed. Either they would be beneficially and economically used for the extraction of metal or not. This, when we talk about the metal source, and as we know very well that the development of any country depend upon the use of metals, resources of metals, and how nicely and scientifically these metals are used. So the primary and secondary natural source of these metals are minerals, ore, and secondary sources, concentrates and tailing. Because minerals you can get in a nature rock, from that you are mining the ore, and for some chemical process, metallurgical process, wherever required, we are concentrating the metal ions and we get the product, that is secondary product, which is known as a concentrates. And from concentrates, by using pyrometallurgy, hydrometallurgy, or chemical process, metals are extracted and remaining material which is produced, which is known as the tailing. At the same time, since last more than 20-25 years, the new terminology has come into the existence, and that is known as urban mining. Normally mine sites, they are in the rural area, near the mountains and all these things, but urban mining, because these are also a rich source of a metal ions, which includes municipal solid waste. It also includes industrial waste, many metallurgical industry, metal industries, and many more industry. Their solid waste is containing variety of metal ions, used catalysts, which are used in the industries, and in the later 21st century and 22nd century, now e-waste, would be a major source of metal ions. So now these metal ions are present either in a primary source, secondary natural source, or in urban mining, either in the form of sulfides of the metals, carbonates of the metals, oxides of the metals, or metal itself in the form of copper or zinc or lead. So they may be present in the form of the compound like a copper sulfide, zinc sulfide, lead sulfides, or copper carbonate or lead carbonate or oxides, or they may be present particularly in case of the electronic waste as a copper ion or zinc iron or gold ion. So from all these sources, how the metal can be extracted, metal can be leached by using the microorganisms, and which is a need of the day, because the sources are depleting, and due to this, the conventional methods which are used for the extraction of metal ion, which is known as pyrometallurgy and hydrometallurgical process. I tried to show you the two pictures. Pyrometallurgical methods, this is a ground method where you can collect the minerals, the underground mines and open cast mine, and you can see that huge amount of the metals in the form of the ores are collected. Here you can see the stacks. Now the depth of this minings are in terms of a few hundred meters. I will visited the many mines which are nearly 400, 500 meter depth, and these stacks are there. So the upper portion of any rock, upper portion of any place from where the mining is done, normally which is not having the metal ions, when you go deeper, then you will get some of the rock which is having the less amount of the metal ions, then you have to go further deep. So when you are excavating all these things, mining all these things, the million tons of a ore, which is a low grade ore, which is dumped surrounding the mine and which is not economically extracted by pyrometallurgy or hydrometallurgical means. I'm not going to discuss about what is pyrometallurgy, what is hydrometallurgy. Just I will give you the brief idea that in case of pyrometallurgy, this ore are heated at 1000 degree to 1200 degree centigrade temperatures, and metal ion are extracted from this. In case of hydrometallurgy, the concentrated acids are used for the extraction of metal ions. Now directly I will come to the subject of the interest of the microbiologist or biotechnologist or the biologist, that is biometallurgy or bioextractive metallurgy, or it is biogeohydrotechnology. Now various terminology are used, but I will select the simplest terminology. Metallurgy means it deals with either removal of metal ion, extraction of metal ion, that is either mobilization of metal ion or immobilization of metal ion or recovery of metal ions. Today, I am going to discuss only one part of this biometallurgy, and that is bioleaching. I'm not going to touch about the biosorptions, which is a biometallurgy of a removal of metal ions from a aqueous solutions or from a solutions. So in case of bioleaching, we are concentrating mainly on the sulfides minerals, because most of the metal ions, they are present at the metal sulfides, and some of the places, some of the metals, they are present in the metal carbonates or metal oxides, but majority of the metal productions is from sulfidic mines. Of course, during my PhD and after my PhD also, I have worked with a oxides course for the extraction of metal ions, but it is not possible to discuss everything in one lecture. So I will be concentrating more on the sulfidic mine. I will give you some overview regarding the oxides and carbonate ores and minerals. Now, if we come more closer to the field where these microorganisms are working, that field is known as biohydrometallurgy. Because as in previous slide, I have discussed with the pyrometallurgy and hydrometallurgy. So it is hydrometallurgy we are using as a base. But in case of only hydrometallurgy, highly concentrated acids are used, while in case of biohydrometallurgy, hardly we are adding the concentrated acids or the dilute acids to maintain the pH. But during the process, microorganisms, they are used in this process. Either they are producing organic acids or inorganic acids, or they are producing some other redox chemicals. So biohydrometallurgy is a branch of technology, branch of biotechnology, which deals with the study and applications of economic potential of the interactions between microbial world and the mineral kingdom. It could be and the metal containing resources. Either it could be ore, it could be tailing, it could be concentrate, it could be a industrial sludge, even the domestic sludge, or it can be a electrical and electronic equipments which are not further in use. And now we come to a biohydrotechnology, to a one closer area of a microbial leaching. So microbial leaching is known as conversion of a water insoluble, or we can say immobilized, metal ions or metal sulfides, or even the metals into water soluble, or we can say mobilized form, metal sulfides using microorganisms or their metabolites. So we are using either organisms or we may be using their metabolites depending upon a single phase process or the biphasic process. In case of biphasic process, we may be separating the organisms and we may be using their metabolites. Metal can be extracted from low-grade ore, from high-grade ores, from concentrates, sludge, as I mentioned, and they can be extracted from the electronic waste. Now, why we require the bioleaching? Now, the question comes the sustainability, and the question comes about the green technology. Some aspects of a green technology, as I mentioned in my previous slides. In case of a pyrometallurgy and hydrometallurgy, we are using extreme high temperature and extreme concentrated acids. And the byproduct produced, they are very, very much, or we can say extremely harmful to the environment. But when we are using bioleaching, we are working mostly at ambient temperature and pressures with a very, very mild requirement of acids. And due to this, the process become a green technology, and the byproduct produced, they can be recycled, and you can have the cyclic process or a loop process where waste are minimum or sometime maybe zero. The other requirement of the bioleaching process are, as we know that the human beings are the most greedy living creature on the world. And each country or even the continent, they want to progress. And as I mentioned in one of my slides, that the progress is not possible without the use of metal ions. And when you want to use the metal ion, you have to produce the metal ions. And for this, you have to use the natural resources. So day by day, natural resources are depleting, and we have to be very careful. Otherwise, after 20-25 years, we may not get the natural resources, because for the formation of natural resources, it takes thousands of years, and for mining, we take few weeks or few months to mine them. The second point why we require the bioleaching is, whenever you are mining, nearly 80 to 90% of the material which are having the low grade ores, which cannot be mined, which cannot be used economically by pyrometallurgy and hydrometallurgical methods. So if you go to any mining sites, you will get piling of a low grade minerals or a dump, and this dump may be in terms of a million tons of a low grade ore are dumped surrounding the mine sites. Accumulation of a huge amount of the electronic waste that is in this 21st century. Now, that I am not going to discuss much about this, but it is in terms of a million tons of a e-waste is accumulated worldwide. Generation of a metal containing sludge, and all this creating serious environmental problems.
Hello, please mute your mics. Please mute.
Somebody is playing the, I think, radio or something. Okay.
Yes.
No? So I am showing with a 2 or 3 slides what is happening. No, this is one of the slides at a sulfidic mines, where you will find accumulation of water which is also in a millions gallon. And the pH of this water, here is sulfidic mine, here is the lignite mines. Now these are the mine where I have visited, and we have published the papers, and we have monitored this acid production for not nearly for 15 years. And every time post monsoons, million liters of the fine drainage waters are collected and having the pH sometime 2 or below 2. So such a huge quantity of this, that is another problem. Of course, this problem is also created by microorganisms. But if you control these microorganisms, then the same microorganisms can be used for the extraction of metal ions. There's another examples. Here also you can find that minerals are there, and copper and iron, and here the ferric iron precipitated, and here you will find the different grade of this metal ion presence. So how the acids are produced? Here you can see in most of the reactions, the acids are produced. The pyrite, in almost all the sulfidic mines, they have the presence of pyrites. And even in the natural ecosystems, bacteria will work on them, and in the presence of oxygen and water is available. That's why I told you that during the monsoon and post-monsoon, the water will percolate. Along with that, you will get some oxygens, and the organisms which are naturally present there, they will convert that pyrite into the ferrous iron and sulfuric acids. The ferric iron, the ferrous will be once again oxidized by the microorganisms, and ferric is produced. Then the ferric iron will get the precipitated ferric hydroxides, and once again the acid is produced. So overall, if you take a 1 molecule of 1 mole of pyrite, will give 16 mole of acids, and such a huge quantity of acids is responsible for the environmental pollutions. But if you use these mechanisms, what microorganisms are doing, then you can use this product, the produce H2SO4 and produce the ferric for the extraction of any metal which is present in the ore concentrate or in e-waste or any sulfidic minerals or even the municipal waste. This now, as I have taken the slides from a Gujarati newspaper, so please just no, it's in Gujarati, but I will tell you. No, this is the one of the very old mine of Asia, which is present in our India, and which is 26 years old. Now, which is more than 100 years old, and in 26 years, it forms 3-kilometer long river-like structures, and it is flowing river where you will find presence of copper, presence of silver, and presence trace amount of the gold and huge quantity of the ferric iron. And this will give you the idea now how much pollution it creates. But if you harvest them and if you use this, then you will get in terms of thousands of rupees of the metal resources. Now I will come to the types of microorganisms which are playing important role here. Chemolithotropic organisms, many of them which includes iron and sulfur oxidizers. And the nature of this microorganism, what we are using, many of them, they are acidophilic, but some of them are the neutrophilic. And we are using these organisms depending upon where we want to use these organisms. Some of these organisms, they are mesophilics. Some of them are thermophilics. You will get archaea. You will also have the role of heterotrophs in the metal generation, metal extractions. If we go to the further diversity, so iron and sulfur oxidizers, which are the main players for the extraction of the metal iron from the sulfide minerals. They are cyclotolerant, mesophilic, thermotolerant, extremely thermophiles, and these are the example of this. Now, this is some of the pictures. No, it is very difficult to grow these organisms. No, Thiobacillus thioparus can be grown because it is a neutrophilic organism where we can grow them around pH 6 or 7. But Acidithiobacillus thioxidans, Thiobacillus ferroxidans, Thiobacillus thioxidans, no, they are fastidious autotrophs, and even the trace amount of organic matter, no, inhibit them. So I have taken some of the pictures from the books, some of the pictures from our publications, and these are the pictures what we have cultivated this organism in our laboratory after many years of trying, and that we'll discuss in some other topics, how to grow these organisms on a solid media and to purify them. Some of the heterotrophic organisms which are also playing important role, and due by the production of organic acids, they are, some of them are bacteria, fungi like Bacillus species, Saccharomyces, Aspergillus, Penicillium, and mainly they are producing organic acid like citrate, gluconate, oxalates. And these organisms are mainly responsible for the extraction of metal ions from oxides and carbonate ores. But at the same time, this Penicillium simplicium and some of the other organisms which are also used for the extraction of precious metal, rare earth elements, and they are also responsible for the extraction of gold. The cyanogenic organisms which are playing very, very significant role, because these organisms are producing HCN, which is the main chemical used by the mining people in the industry to extract gold. But they are using cyanides. That itself is, we know that cyanide is such a toxic compound and which create a lot of environmental problem. While these are some of the organisms what we have used in our laboratory and the HCN productions, and we have used them for the extraction of gold and silver. Now this is the reactions, how the silver, the cyanide which is produced by microorganism, now they will make the complex with a gold and you can solubilize the gold. Now coming to the next topic of this proposed mechanism of bioleaching. Why I use the word proposed mechanisms? When I was doing my PhD, now I have started my PhD in 1977, and I am fortunate to get the PhD on bioleaching first in India. At that time, I know the three peoples, they were working some aspects of this bioleaching. One is Dr. Akte, Dr. K. Natarajan, my immediate supervisor and my guide for my PhD, and Dr. Saroj Mishra at Dhanbad. Now they have worked, or they have slightly worked as because they are related to this mining industry. And my guide, Professor K. Natarajan, he was there in Minnesota University, and he is the extractive metallurgist. But there in Minnesota, people were thinking about the use of microorganisms, and he came to India, to KREC, and I was fortunate to get selected for my PhD in the engineering college. So he was having the background of extractive metallurgy, and I am having some knowledge about the microorganisms. And my guide, Dr. J.V. Bhatt, the father of microbiology of India, he told me that the way I don't know anything about the bioleaching. Dr. Natarajan also told that I will guide you as far as the extractive metallurgy is concerned, but you have to develop. And at that time, that I am talking about 1977 to 1989, 200, now this bioleaching process was classified as direct and indirect leaching. Thereafter, as the field is developed, and when we'll discuss this process, we will find many things overlapping, but the terminology has changed. People have used, instead of direct and indirect leaching process or mechanisms, the new terminology came into the existence, and to use a contact mechanisms, non-contact mechanisms, cooperative mechanisms. And thereafter, in the 21st century, even these concepts are slightly modified in terms of the biochemical reactions. Now, what you are finding in terms of acidolysis, redoxolysis, complexolysis. So now all this process, all these mechanisms, no, I cannot say that as I am working in this field since last 45 years. So there is a lot of overlapping, and people have tried to explain them on the basis of the particular concepts. No, if we, as I said, that the iron oxidizing organisms, Acidithiobacillus, Lactospirillum, Aeromicrobium. Now we are working with these organisms, and we are having the consortia of this. And this all of you, those who are working in biology, they know the 9K medium. That Silverman and Lundgren, they have prepared the medium. And once again, I say I'm very much fortunate that I have spent a few days to few weeks with a Tolward, those who have worked on the bioleaching globally, and not only the academically, but they are the in charge of a mining industry, and they were involved, such as Silverman and Lundgren, who has first isolated Thiobacillus ferrooxidans, and I was fortunate to work with them. Then Tobinen, Torma, Gurudev, and many more. No, they are all in charge, and they are working. They are producing a million. They are working with 1 million tons of ore, and actually they are producing. And so here this mechanism, as I said, the ferrous would be converted to ferric, and this ferric will react with the metal sulfide, any metal sulfides, and they will produce the metal sulfate. The one of the example I have taken with the copper minerals. Copper minerals, it react with a Fe2SO4 thrice, that is the ferric sulfate, and you will get copper sulfate. And most of the metal sulfate, except the lead sulfate and barium sulfate, no, they are water soluble, so you can extract metal ions. The other main source of this production of H2SO4, that is the sulfur or thiosulfate or sulfides, where sulfur or sulfides are acted upon by the microorganisms, Acidithiobacillus thioxidans or sulfolobus, and H2SO4 produce. And this H2SO4 react with the metal sulfides, and you will get metal sulfate in soluble form, and sulfur once again. Because sulfur once again oxidize. What I said, it's a by green technology. The product sulfur, it is produced once again oxidized by this microorganism. No, a ferrous which is produced once again oxidized by this microorganism. So it is a cyclic process. Electrochemical reactions are also taking place, where The ferric sulfate is similar one, but the electrochemically, because when you are working with the minerals, when you are working with the ores and concentrates, the galvanic interactions, it is bit complicated reactions. Wherever if copper, chalcopyrite and covalite are present, the covalite will solubilize first as compared to chalcopyrite. But if the chalcopyrite and pyrite is present, chalcopyrite will be solubilized first as compared to pyrite. No, this will discuss some other times. So now this is the electrochemical mechanisms. This bacterial cell, whatever it may be, either it is acid thiobacillus thioxidants, acid thiobacillus thioxidants, or acid lactospirillum thioxidants. Now this APS layer, and in APS layers, now it is producing ferric sulfate, and that ferric sulfate is reacting with the minerals, not necessarily only the pyrite, and it is solubilizing the form of sulfates. Now this, as I said in that one, the non-contact mechanisms, contact mechanisms, cooperative mechanisms. In non-contact mechanism, ferrous or ferric is produced, and without the contact of microorganisms, they are solubilizing. In contact mechanisms, the bacterial cell, they are coming in contact with the minerals, and during this EPS layers, the reaction is taking place, and combination of this, that is cooperative mechanisms, and you will get the extraction of metals. Commercial processes which are used for the extraction of metal ions worldwide. Unfortunately, in India, still we are not successful to convince the mining people that you have to use this process. It is a slope leaching, heap leaching, in situ leaching, dump leaching, and the reactor leaching. I will show you the combined figure as well as the individual figures or individual pictures, how to explain this. Here you can find the slope leaching, or it may be known as a dump leaching also. Then here the heap leaching and in situ leaching. Now I will show you the bigger figure of this. Dump or slope leaching. Here, as I showed you in some of the pictures, that when you are mining, low grade ores and high grade ores, which is if you just dump on a mining site and on a slope, then it is known as a slope leaching or a dump leaching. When smaller amount of this material, and we can have the percolators, and we grind the materials and have some combinations, it is known as heap leaching. In situ leachings, where it is not economical to mine the minerals, the cost of the mining will be more than we are going for the in situ leachings. Then you have to study the geological cycles, and you have to study the hydraulic cycles which is flowing, and accordingly we have to do it. This is, I said, the chalcopyrite, covalite, chalcosites, some of the copper minerals, and you will find here, this is pyrite, and this is the presence of coppers. Now this is presence in case of covalites and all these things. Once again, pyrite. So galvanic interaction are playing. It is a very complex process. Because in the presence of covalite, chalcopyrite will not get leached out. But in the presence of pyrite, chalcopyrite can get leached out. All three are the combination of a copper metals. Bioleaching of coppers can be, this is one, and 25% of a world production, 25% of a world copper production is by the bioleaching process. Now, it is not only these two organisms, because this process we are doing in terms of 1 million tons of ore. So it is not a single organisms, it is a consortia and natural organisms which are present, but there are few tens of the organisms which are playing very dominant role. We will not go into the much detail, but this will give you the idea how the chalcopyrite will get copper, how the covalite will be converted into a copper sulfate. This chalcopyrite also copper sulfate, and chalcosite will also convert in copper sulfate. And this will show you how much amount of the acid is produced. Here the 8 moles of acid is produced. Here 2 moles of acids are produced. Here also some of the acids are required. Now this is a Chile, which is having the large production of copper, And no, this is how much copper sulfate they are able to leach out. You can see it is in terms of a millions gallon of the copper sulfate is leached out on the mining sites by these organisms. It won't require much a sophisticated fermenter what we are using in the pharmaceutical industry. Now this will give you the idea. The productions of a various metals, tons per day. It is not tons per year, it is tons per day, 16,000, 16,000, 2,000, 17,000, 32,000. And there is various mines at the various places. Now, I have started my PhD in 1977 with work with a sphalerite concentrate in Kobinko Minani company. Now, we got the sphalerite concentrates and I have developed that process at that time to a 25 liters and more than 95% of the zinc extractions by using at that time the organisms what we were using was known as Thiobacillus ferrooxidans, and in 2003 it renamed and it is named as a acidic Thiobacillus. So I have worked with even the chalcopyrite ores, the reactor leaching, no, that I showed, no, either third tank or elliptic tank, or no, this mainly used for a very high grade ore or a concentrate. The store tank bioleaching operations. Now this is also they are producing tons per meter cubes per day. Now arsenic and arsenopyrites, pyrohortites, complex ore containing copper, zinc, lead, then stepnites, pyrohortites, various country. Unfortunately, we cannot see anywhere name of our country, and we think that we are not able to convince our country people. This is a cobalt leaching, or Kesa Cobalt Uganda, and they are producing 1,000 tons per year of a cobalt by the reactor leaching. Now, as I told you, there are some of the minor ore which are having the either carbonate or oxidized ore, where also we can use various types of the organisms. And in 2021, leptospirillum periphylium is used for the production of metal ions. 2 or 3 of my students, we have used leptospirillum periphylium dominated cultures, and we have published paper in 2012, and from more than 2012, we are working with the extraction of copper, zinc, and lead from a complex ore materials. We have developed the process where we can go up to the 20-25% pulp density. At present, also at many places in India, they are working with a 5% pulp density. One of my colleague, Dr. Devyani Tipre, she has also worked on the Amba Mata complex ore, and we have extracted copper, zinc, and lead from the ore and concentrates. She got her PhD, and more than 5-6 students, they got their PhD under me on the extraction of metals from the complex ores, concentrates. Even in case of the pyrites, oxides, carbonates, other than the Thiobacillus and leptospirillum, no, Aspergillus nigers. I have also used Aspergillus nigers, Penicillium sicilium, and Pasolomyces for the extraction of copper from the oxide source. But all these are for before 1982. So I have not put any of this reference here. Then some of worker in our lab, some of the PhD student, Monal, she has published 3-4 papers in 2014-15. Asha Soda, she has published papers from 2017-18. Bhumika, she has published few papers in 2019. Seetal, very recently, she got her PhD in 2021. And Apilas, we are having the collaborations at NMS. So this oxide source and uranium, where we can leach out this uranium. We are not going to detail about this. Electronic waste, also I'm not going to much detail because I'm about to have the 40 minutes. So, but in case of this, printed circuit boards, In last 10 years, we are working on the electronic waste, and we have developed the process where we can extract metal ion, more than 90% metal ions, and that too within a 4 or 5 days, while people are requiring till 20-25 days, where our process and Seetal and the other research fellow, those who have, they have also extracted around 50% of the gold from electronic waste. Here, there are many problems. I have not, as I have discussed, I am not going to discuss about the various problems which are affecting factors. But in case of electronic waste, addition of the material at times, mixed inoculums, and this process are playing very crucial and significant role of this. Biology research in India, IISc Bangalore, where my guide, Professor Natarajan, is still working. No, he is around 80, but in IISc, no, they are fortunate to continue their lab, and almost every year I am spending 1 or 2 weeks with him and still working. Then RR Bhubaneswar, they are working with the dump and heap leachings. School of Mines, Dhanbad, NML Jamshedpur. We have seen the one of the paper of the Abhilash. Now we are having collaboration. Agarkar Research Institute, Pune, KRAC, where I was working, and we have developed the process, and Department of Microbiology, where I was working, and still I am having some of the students working under Dr. Devyani Tipre, and we are continuing this process for the extraction of copper, zinc, lead, gold from concentrate, from ore, from tailings, and from e-waste. Now, what are the advantages of this process? It's a simple and effective process. You can work with a low grades, you can work with a waste, you can work with a concentrate. Mainly, you can work with ambient temperature and pressures, which is very cost effective. Now, we have done the calculation also from a concentrate leaching, and that copper productions would be 17% cheaper as compared to the existing cost and existing process. And upgrading of the minerals in case of the, it is less energy demands. It is very environmental friendly because we are not using any very harmful chemicals. We are not using very concentrated acid or alkali. Flexible for the treatment of minerals resources with a variety of metals and at a variable concentrations. And you can get the productions now within an acceptable period of time. Limitations. In terms of a mining people, they say it's a slow growth rate. And of course, as compared to the pyrometallurgy and hydrometallurgy, the process is slow growth. But when you talk about the world production of gold and copper, as I said, in copper, it is 25% of the world copper production is by this method, and nearly 23% of the world gold production is by this method. And if this process are economical in any foreign country, it would be at least 1.5 times more economic in our country. And that is due to the what gift we are having, natural gift. Now, in our country, most of the time we have the temperature above 30 degrees centigrade, while most of the other country, most of the time temperature is below 25. And so there they have to use more heat for this, or the rate of reaction would be at least 1.5 times slow as compared to rate of reaction we'll get in our country. The other problem is metal tolerance. But if you are working, we are having the organisms which can tolerate more than 14 or 15 metals, even in not in terms of a ppm, but it is in terms of a gram percent metal tolerance we have reached. So there is not a question, only you have to develop and you have to adopt these organisms. I want to thank my guide J.V. Bhatt and Dr. Natarajan. Then this work I have done on the sphalerite concentrates and some of the ore. Rest of the work, what I have presented here and what I am dealing with now, it is Dr. Ajit Menon, D.R. Tipre, B.C. Bhargav Patel, M.J. Patel, M.B. Shah, A.B. Soda, Seetal, and Bhumika. So, and I also acknowledge the various funding agencies, those who have given me the funds to carry out my works. So this is one aspect, a very, very brief idea of how the leaching can be done, how it is sustainable, how it is, we can say, green technology. Thank you very much. And if you have any questions, I will be very happy to answer. And once again, I want to thank the Microbiological Society of India for giving me this opportunity. Thank you very much.
Thank you. Now we have time for some questions. Anybody can ask. The students, you should ask some questions from Dr. Dave. Dr. Dave, I'll ask you a couple of questions.
Okay, okay.
Which are the microbes you used mostly for bioleaching, number one? And how were they preserved for a long time?
Okay. So the one we are using, if you are using the sulfide ores or a electronic waste, then mainly we are working with a acidithiobacillus thioxidants, acidithiobacillus peroxidants, leptospirillum peroxidants, and other iron and sulfur oxidizers. If we want to extract precious metal and rare earth metal, then we are using some of the heterotrophs, and you are the expertise of preserving the heterotrophic organisms. But to preserve these autotrophs, iron and sulfur oxidizers, because they can be preserved only in the liquid medium.
Yes.
So for a long-term preservation, what we are doing, we are taking a low-grade ore. We are taking a low-grade ore, and then as we are preparing a biofertilizer, similar way we are putting these organisms on a low-grade ores and maintaining the just require the humidity or the moistures. And I have found that this organism remain active at least for 5-6 years without any transformations, any transferring, and till no. I will give you one of the examples. No, I have collected one sample from mining, and that sample mine drainage we have kept, and every 2 years I was examining them, and we got the leptospirillum peroxidants, iron oxidizers, even after 15 years.
Yes.
But that is not preserved in the laboratory. That mining samples I have kept when I have isolated organism, and that samples I have kept, and after 2-3 years, just I was, for curiosity, I have tried to grow. I got them. Then I have preserved in a room temperatures. So in a nature, these organisms will survive for years together. If we try to preserve them in a laboratory conditions, you have to preserve only in a liquid or by taking a very low grade ore. So they will get their food, but not as much as that the metabolite will be inhibiting this. So this is the way we are preserving.
You know, the pH is very acidic. It is 3 to 4 in case of thiobacillus. So we cannot store in agar. We cannot store in -80.
Yes.
It is difficult to lyophilize also.
Yes, yes.
So the best way is liquid cultures.
Yes.
This is a message for the students that thiobacillus, sulfolobus, etc. are always stored in liquid medium. So number 2, did you use surfactants or rather biosurfactants for bioleaching?
Means we have not used biosurfactants directly, but when we want to optimize the process and to make the process much of faster, then we have used some of the surfactants, chemical surfactant and biosurfactant, which playing a physico-chemical interactions with minerals, and which make more bioavailability of that minerals to the organisms. And we have used this particularly in the electronic waste, and where we have got one of my student, Monal Shah, she has used 4 or 5 this type of the biosurfactant and chemical surfactants, and we have got, I think, 30% more extractions at a faster rate.
Yes, actually biosurfactants, they mobilize the metal.
Yes.
And especially in precious metals like gold, platinum, they are very, very useful.
We have used even the EDDA, and that has given very good improvement in the metal leaching.
Right. Yes, thank you. Please ask question.
There are few questions in the chat box.
Please read.
Read them, sir. Should I read these questions?
Yes, please.
Okay.
So one question is from Srirang Madal. How much scalable is this technology for the recovery of metals from e-waste? And he's also asked how much commercially viable is it to set a microbial recovery plant, metal recovery plant with this technology?
In case of e-waste, we have developed up to a 30-liter reactors because no, we want to make this process much faster. So we are working with a reactors, and there within 3 days we are able to extract more than 90% of this. And when we talk about the electronic waste, so we have to see on both the sides. It's the environmental pollutions and the metal recovery. If we recover along with the copper, if gold is also present, if we recover gold, then it is economically viable. Only the question comes that somebody should come forward to work with a electronic waste. We have worked with a mobile, then laptop, computers. We have worked with even the radio, TV. We have worked with electrical equipments almost every time. And that we have developed a process where if it is a bilayer or the monolayer of the PCB is there, you should not grind. We were also grinding before 10 years, but grinding cost will increase, pollution will increase, the volume of the material what we are using, that will also be increased, and we have developed the process, but that is useful for a monolayer metal PCB and the bilayer metal PCB, and that will not give any environmental pollutions, and that fiberglass or the plastics which is used, that can be reused for the other purpose. So this is the answer. And second question. Yes, no, the second question is that bioleaching copper. One of my students, no, we have got this copper sulfate produced by this organism in a nanoparticles, but we have not further, no, because once they will get the PhD, they will not continue. But we have developed and we have observed the nanoparticle formations during the leaching period. Not only the copper, but we get the iron nanoparticle also. And there is second line, that this biological process can be developed for the production of a colored material from the iron. I have worked with a production of a yellow color, then a red color, then brown color, and which have very good market.
Any more question?
Sir, there is another question in the chat box. Are there any existing plants for metal recovery from e-waste with this technology in India?
No, at present, I think in Maharashtra, I come to know that one person has started, but laboratory level, we are having the know-how. If anybody is interested, because we work with all types of electronic waste, we have the culture which can grow up to a 20% of pulp density, either it is a pulverized e-waste or unpulverized e-waste. We have developed a culture which can produce ferric, which is the main leaching agent for electronic waste, very, very fast. Even our organisms are able to do their function at pH 1 and in presence of many metal ions. So if anybody is interested to scale up this process, and we have also developed the recovery process. Of course, recovery process we have developed at a small scale, but recovery process you will get in any metallurgical laboratory. It remains one and the same, either your leachate is from an electronic waste or a minerals.
If there is no more question.
Sir, I have a question.
Yes.
Sir, like if we are using phytoremediation, so are these plants, are these plants can be used for extracting the metal by bioleaching? Is it feasible?
No, this phytoremediations mainly used
No,
For
sir.
the-
I'm asking. My question is, I am using phytoremediation, and these plants are with heavy metals. Plants are taking the metals, whichever metals.
Yes.
So, is it possible to use these plants for, I mean, by bioleaching for extraction of metals?
No, that's what I'm answering. This
Hmm.
plant can be used, in one of the slide I showed, the biometallurgy, one is leaching, other is the absorption, biosorption. So the phytoremediations are used for a metal containing liquid waste. No, then you can recover the metals. We have developed one of the fungus culture, Aspergillus terrace. No, the beauty of that culture is it is selectively removing the gold and platinum and silver. While most of the other plant material or fungi, no, they are not much selective, but it cannot be used for bioleaching. For bioleaching purpose, no, you have to produce either the inorganic acids or organic acids or ferric iron. So this phytoremediation is used for the cleaning of the acid mine drainage or the industrial waste pollutants or even the any water body which is contaminated with this.
Mm-hmm.
Dr. Pachankar, you have any question? All right. It was a wonderful talk.
Yeah, it's a wonderful talk.
Wonderful talk. Yeah. We are very happy.
Actually,
and we have learned so many things.
near Nagpur, Manganese Ore India Limited is there, and they are doing manganese extraction by bioleaching.
Yes.
They are having a good plant.
Yes.
No, we have also tried to set up one plant for copper extraction at Malankhand mine, and one plant demonstration plant we have set up at the Hatti gold mine.
Malachkhand is also there, sir.
But
Yeah.
Unfortunately, they are not continuing. No, every time when we contact, they say you purchase the mineral and you start. No, we are academicians, we are scientists. It is even very near to Gujarat. We have the Zawar mine, in Udaipur. There they have the problems zinc, and we have worked with that zinc tailing. We have worked with my PhD in the zinc sulfides, but they are not coming for. They say no, let the waste would be accumulated. That we'll see after 10 years, 15 years. Now this is why the India is lagging behind as compared to many countries. No, all other countries, they are using 25% of copper and 23% of gold. It means they are saving 25% of their mineral every year.
Yes. Dr. Acharya? Dr. Acharya, you have any question?
No, sir. Right now, I do not have any question, because if there is a question, I will direct contact with the sir. Because
Yes.
I am constantly in contact with the sir,
Very
because
good.
he is my gurus and the mentor. So I bow my head over here on the auspicious.
No, but you can ask question. No, you can. If you have
Right
any
now, I
question,
do
you
not.
can ask. No problem.
Because right now I
Yes.
am not, sir, working on the bioleaching
Okay.
aspect.
Acharya,
Yes,
this is a
yes,
good
sir.
moment to say Guru Purnima to your.
Right. Thank you.
I'm
Thank you,
not.
sir.
I'm very happy that on Guru Purnima I got this chance to give some of after retirement. This is main motto is to serve the student community and the subject people, and this is the opportunity. I'm very much thankful to the society and the
Very good.
Suranjit Singh for inviting me and giving me this opportunity, because
Thank you.
I want to develop this field. Now that is the reason. Many people have told many times why you are not going to any lucrative field of microbiology. I say that I have done my PhD and I have promised Dr. J.V. Bhatt and Dr. Natarajan that in biological field, as long as I will able to do work, I will continue with bioleachings and the metals.
Very good. Thank you, sir. And Kulveer, you can do your next event.
Thank you very much.
Thank you.
We had an amazing session today. On behalf of the organizing team, it's my privilege to propose a vote of thanks after this wonderful interactive session. My heart fills with gratitude and a lot of respect for our distinguished Professor Dr. S.R. Dave, who not only spared his valuable time to be with us, but also enlightening us with his commendable knowledge about this sustainable green solution in biohydrometallurgy. And his field of work is amazing. So we are highly obliged. My humble thanks to the president of the Society of Microbiology Society India, Dr. A.M. Deshmukh, for his commendable enriching support always. To Dr. Swaranjit Singh, our respected director from SAS Polyclinic and a reputed microbiologist, a very kind human being, for his love and support always. I'm also thankful to Dr. Patnakar for being with us and his support always. My gratitude to the technical team for conducting this webinar smoothly. I must mention a deep sense of appreciation for all the members of Microbiology Society India, all the coordinators who are working hard for creating platforms for sharing this microbiology knowledge. And in the end, all the lovely participants for this webinar, because without them, this would not have been a better session. Thank you all. Thank you all. Good day and kind regards to all. Thank you.
Thank you.
Thank you, Kulveer,
Thank
for
you.
the nice vote of thanks.
Thank you, sir.
Okay, bye.
Bye.